Hello and welcome back. Biology for Beginners is finally returning from its brief hiatus when I basically couldn't be bothered to open my Kindle app because 2020 is confusing. And if you want a little bit more context on that, please go back and watch my last video all about the duck situation. But without further ado, welcome back to Biology for Beginners. As always, I am your host, Morgan. This week we are finally beginning something that is really within my wheelhouse and not scraping up my shaky knowledge of biochemistry and organic chemistry. We are diving into chapter 4 and talking about cells. So this week our topic is a brief introduction to cells. As always, at this moment in the video, we are showing our terms and definitions slide that are full of all the fun sciencey terms you will be hearing throughout this video. If you would like, please feel free to pause and grab screenshots of those slides. Or you can pop on over to my Patreon page and grab my entire slideshow as a PDF for absolutely free. This week, there will also be a couple of other things going along with this slideshow that will also be in that Patreon post because of some comparisons we will be making to different types of cells, and that will be a theme in this particular chapter as we go along. There will be both the filled out version of these charts and a blank version if you would like to grab that and fill it out yourself and use it for any studying help you might need in any biology course you are taking right now. So without further ado, let's begin. So before we really get into the nitty-gritty of cells, first let's look at the tools we need to be able to even view cells. And the first tool we're going to look at is the most basic light microscope. These are the microscopes that you may remember using in your middle school or high school biology classes and may currently be using now if you are in middle school or high school or in a college biology class. These microscopes were first used in the 1600s and can magnify things up to about 1,000 times. They work by passing visible light through a very thin specimen and projecting that image onto your eye, or more modern ones will project it onto some sort of photo medium or even into a computer. Light microscopes are super cool and I am obsessed with them because I am a nerd. The other thing I would really like to point out on this slide that we are seeing, if you look at that lovely little paramecium right there, the red circle to the side of it with the LN230X is showing the kind of microscope and how much the particular image is magnified. And that is something that you will see no matter what kind of microscopic photo you are looking at. Here it means it was a light microscope magnified at the 230 times magnification. So LM, light microscope. And now to move on to our next ways to view cells. Now, light microscopes are wonderful and they completely catapulted the biological sciences forward in the 1600s. But they're only good for things that are a very specific smallness. So in the late 1950s, we began using what is known as an electron microscope. And what these do is they focus a beam of electrons specifically to look at the surface or the interior of whatever specimen happens to be there. And instead of glass lenses, they use electromagnets so that those electron beams can be focused. Now, there are two kinds of electron microscopes we are going to talk about right now. The scanning electron microscope, which is used to create a 3D image of the surface of a specimen, and the transmission electron microscope, which is used to study the internal structure. Now, in a very basic biology classroom, you're not going to have access to electron microscopes, and truthfully, I've never seen one in person. But in textbooks, you will often see these very close up and interesting images like we have here in the slide, showing very detailed looks at the surface and interior of these specimens. And again, these images can be colorized or not, and there's different ways of staining and other computer programs that go in to using these specific kinds of microscopes. But that is about what you need to know about how they work. And the big difference between the two kinds of electron microscope is just how the electron beam is focused and what they're specifically looking at. You may be sitting here thinking, well, why do we need such specialized tools to look at cells? Well, it has to do with their size, and we're just going to take a little brief pause here to talk a little bit about the size, scale, and kind of measurement of everyone here. So if you look at the images I have on this slide, it's a very simple table of the very basic measurements that you will see in any kind of science textbook, specifically with the meter, centimeter, millimeter. Metric system is the system used in science. Metric system is very logical because everything's just a tenfold step. And you can get from like nanometer to meter very, very easily just using multiples of 10. And basically what I'm saying here is the measurement system we use in the United States is complete BS. I hate it, and we should be on the metric system with the rest of the freaking world. But that's my rant for the day. So directing your attention from the metric measurement equivalents that I'm truthfully not really going to walk you through right now because it's not relevant to anything else I'm going to be talking about. I just want you to be able to see it. And if you have a question about it, please feel free to put it in a comment down below, and I can give you some resources to learning how to better get through all of that. Basically, if you can get meters, grams, whatever, you understand how to go between the different types of one, you understand how to go between the different types of all of them, because it's the same basic system, just with different words, and they're measuring different things. And that's the big deal. I'm not going to be asking you to do math. I'm not that mean. I'll just make you sit here while I geek out over how cool cells are. And I don't know which fate is worse, but I've decided that I'm right, and I mean, you're still watching this video, so that's on you, honey. But if we look at this size chart that kind of shows the scale of everything and the reason we need microscopes, even though with the unaided eye, we can see, we can kind of see a paramecium, which is a microorganism, but it's not going to be detailed, obviously. It's very small. So we use these microscopes to get down to the very small levels and see the details. And without these microscopes, we wouldn't really know that we're all made up of cells, that living things are made up of cells. And we wouldn't have cell theory, which in 1665, Hooke discovered cells. And after that, using these light microscopes, scientists continued to collect information from these microscopic organisms and from tissue samples taken from plants and animals and fungi and all those other fun biological things we have in the world. And once all of this evidence was squished together and the data was amassed, cell theory was really rounded out and put into words. But the basics of it is that all living things are composed of cells, and cells come from other cells. So cells make up everything living, and you need a cell to get another cell. The next question we need to tackle here, now that we kind of know that cells exist and you need a microscope to see most of them is, why the heck are cells so small anyway? What is the possible advantage that can come from having a bunch of very, very tiny cells? Well, this all has to do with the surface to volume ratio. So to put it into some very basic, quick and dirty explanations, the volume in one large cell can be exactly the same as the volume in many small cells, which would mean they can hold the same amount of stuff. However, one large cell has less overall surface area available to transfer materials between other cells. And the many small cells, because they're going to be a bunch of, in the example in the textbook, it's using cubes. So for simplicity, we'll use cubes. A bunch of small cubes, every single one of those has 6 sides. Each of those sides adds to the overall surface area. So that is a lot of surface area available to transfer these molecules and ions between cells. And a very large cell only has the 6 sides in that cell available. So overall, if you have a bunch of large cells versus many, many more small cells in the same volume of space, you get less transfer materials, it's less efficient. So if you have many, many small cells with much more surface area, transfer of materials and by consequence transfer of biological information and resources is much more efficient, much more quick. And overall, that is better for that biological system made up of those cells. I do hope that I wasn't too confusing there, because I do tend to kind of talk when I get excited about things, but feel free to pop a comment down below and I can elaborate on anything you need me to. But the surface area that these cells are using to transfer things is the plasma membrane, or the cell membrane. And what this is a phospholipid bilayer with proteins embedded in it. And this controls the flow of those ions and molecules into and out of the cells, which are the materials being passed from cell to cell. And the image here in this slide is a generalized version of what that phospholipid bilayer with those embedded proteins may look like. But we will get more into the specifics of that later on as we explore cells. Now that we've talked about cell theory and why cells are just so darn small, let's talk about the kinds of cells that you will encounter on planet Earth. The first kind of cell we're going to talk about is the prokaryotic cell. Prokaryotic cells were the first to evolve, and they inhabited the Earth alone for more than 1.5 billion years, meaning for that amount of time, prokaryotic cells were the only life on the planet. Prokaryotic cells are still alive today, and they are represented in the domains bacteria and archaea. These cells are very small and very simple, meaning they have no membrane-bound organelles. So they only have that outer cell membrane. Their little organelles within them are non-existent. There are no membranes within the body of the cell. They don't have a nucleus like the cells we will talk about in the next slide. Instead, their DNA will be concentrated in what is known as a nucleoid. However, that DNA is still in contact with the rest of the stuff in the cell. It is still completely in that cytoplasm, and it is not bound by any kind of membrane. It just happens to be where the DNA is concentrated. Prokaryotic cells have a cell wall, and then they may also have flagellum or flagella, which flagella is just the plural of flagellum. I did not mean to make it sound like they are two different things, but there may be one or many present to help facilitate movement in their usually aqueous environments. So as I implied while we were talking about prokaryotic cells, they were only alone for about 1.5 billion years, which means something else had to evolve. And that something else evolved about 1.8 billion years ago, and they are known now as eukaryotic cells. Eukaryotic cells make up every living thing except for the organisms in domain bacteria and archaea. So an easy way to remember that is that you have eukaryotic cells. Compared to the prokaryotic cells, eukaryotic cells are rather large, and the thing that really defines them is that they contain the membrane-bound organelles like the nucleus and the mitochondria that has been so wonderfully driven into our brain with public education as the powerhouse of the cell, which we will get more into the mitochondria in future videos. But an easy way to remember what organelles is that they can also be analogous to organs in a greater body, and they're technically little organs. And these organelles are specialized for the specific functions of the cell that they are in. And we will get into those specific organelles in future videos. And the other important thing about eukaryotic versus prokaryotic cells is that the DNA is contained in a membrane-bound nucleus in a eukaryotic cell. Whereas, remember, prokaryotic cells have nothing membrane-bound within them. And then eukaryotic cells may or may not have cell walls. The biggest example of the cell wall in eukaryotic cells is that plant cells have a cell wall, whereas animal cells do not. But we've talked about their differences. There are still some things that prokaryotic and eukaryotic cells have in common, and we will get into that right now. The basic things that prokaryotic and eukaryotic cells have in common is that they have a cytosol and cytoplasm, which is basically the inner parts of the cell. And for the purposes of these videos, I use cytosol and cytoplasm almost interchangeably, and in most basic biology, intro-level general biology classes, cytosol and cytoplasm are used interchangeably as well. However, the distinction here is that cytosol specifically refers to the jelly-like fluid within the cell, and the cytoplasm is all of the stuff in the cell. But for the most part, our discussions, if I say cytosol or cytoplasm, I'm just talking about the inner parts of the cell for the most part. I will be specific if I need to be, but I highly doubt that will be necessary for the purposes of these very basic videos. But moving on, prokaryotic and eukaryotic cells both have chromosomes, which are the things that carry the genes made of the DNA that make the cell do the cell thing and make the life do the life thing. And because they have that DNA and chromosomes, they necessarily need to have ribosomes, which are those areas of protein synthesis. And when you're looking at every single thing a cell does and all those chemical reactions that happen within that cellular membrane and cell wall, if the wall is present, but it doesn't necessarily have to be. Cells don't necessarily have to have a wall, just a membrane. And even if you have a cell wall, you will also have a membrane. But I'm getting ahead of myself. Every single cell, if it is still alive, will have a cellular metabolism, which is just basically all of those lovely chemical reactions going on inside the cell in question. All right, well, I've probably tortured you with my rambling about cells and attempting to get ahead of myself enough for one video. I do hope that I didn't scare you too much and you will show up next week for our next leap into the world of cells. but I understand if you run screaming for the hills because I am a little cell enthusiast nutcase. But with that being said, I do hope you have a wonderful rest of your morning, day, evening, night, what have you, and I will see you all in the next one. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane B. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.